Welcome, ladies and gentlemen, to the very next exciting adventure in Unit 2, where we talk about things that limit the size of cells and explain why we don't have cells the size of people or that you can easily see with the naked eye, and also describe some of the basic properties of the cell membrane. So let's get started, shall we? First, we need to think about what a cell needs in order for it to stay alive. We talked a little bit about this when we talked about the needs that all living things have, the characteristics that all living things share. Well, we all need glucose, some sort of energy source. We all need oxygen, which is used to break down the glucose. And we all need nutrients, other molecules that we use to build the molecules that make up our cells. And those needs are no different for individual cells themselves, whether within our body or as an independent organism like a unicellular organism. Similarly, all organisms need to be able to remove waste products, both solid waste products and carbon dioxide, which is a gaseous waste product. So, for every cubic volume of space that you have inside of a cell, you need a certain amount of these materials to enter and exit the cell. By the way, that cube, that nice blue cube in the center, is our kind of model of a cell that we're going to be looking at for the next few slides. So let's talk about how materials enter and exit the cell. They enter and exit the cell through the cell membrane, and I've kind of given you a visualization of the cell membrane here. It's basically, you could pull it off each side of this cube, and so you should kind of see that these 6 squares form the cell membrane of this hypothetical cell that we have here. And each square unit of area, each chunk of this cell membrane can only transport materials at a certain rate. So for the sake of argument, we're going to say that this cell is going to be 1 nanometer in length, width, and height, and that this membrane can transport 1 molecule per hour. These are all hypothetical numbers that I'm mostly using so that we can kind of visualize this, or so that we can get some examples. So what we're going to look at next is how many molecules can this cell, the entirety of this cell membrane, transport within 1 hour. So if we look here, we have this 1 nanometer cell. We have a formula for surface area at 6 times the length of the side squared. And we also have a reminder of how many molecules can be transported for every square nanometer of space. So first, let's calculate the surface area of this cell. It's going to be 6 times that 1 nanometer squared. And we square everything inside of those parentheses, so it's 6 times 1 squared nanometers squared. And 1 squared is 1. Anything times 1 is itself, so this ends up being 6 nanometers squared. Now, since we're calculating all this for an individual hour, I'm going to go ahead and take this off of our units just to make the math easier. So if we're trying to calculate how many molecules this cell can transport within an hour, we're going to multiply the surface area by this number. 1 molecule per nanometer squared. Now, you can treat this as multiplying 2 fractions, and when you multiply fractions, you multiply across the top and across the bottom. So this turns into 6 nanometers squared times 1 molecule divided by nanometer squared. If you haven't done this in chemistry yet, you can do math with units. So for example, I have something on the top of a fraction and the bottom of a fraction, that nanometer squared, meaning I can divide those two out. Anything divided by itself, like say nanometer squared over nanometer squared, equals 1. So I can basically get rid of it because it's just like we said before, multiplying anything times 1 is itself. So this thing can transport 6 molecules per hour. Okay, we're going to keep that in mind for this next part. So this cell requires 5 molecules per cubic nanometer of space so remember it's volume, per hour to survive. So how many molecules does it require in 1 hour? We've got the formula for volume here, and it's got the same length of sides. So let's calculate the volume. The volume is 1 nanometer cubed. We cube everything inside there, so it's 1 cubed. 1 to any power is 1. Nanometer cubed. If it requires 5 molecules for every cubic nanometer, then we can do this math. 5 molecules for every cubic nanometer. Just like I said on the previous slide, you can treat this as multiplying two fractions. You multiply across the top and the bottom, so that becomes 1 nanometer cubed times 5 molecules over nanometer cubed. Sorry, I tend to drop ones when I'm multiplying, since anything multiplied by 1 is itself. As before, I can cross out anything that's on the top of the fraction and the bottom, so that lets me get rid of those nanometers cubed, and so this means that this cell needs 5 molecules per hour in order to survive. Now again, this is a gross simplification, very dramatically simplified, but we can use this to just see the principle. So this cell can transport 6 molecules per hour. It needs 5 molecules per hour. So basically, can this cell survive? It can transport 1 more molecule than it needs, so it is capable of surviving. But now, let's see what happens if we change the size of this cell. Now, instead of being 1 nanometer length and width and height, it's going to be 10 nanometers. So let's calculate the surface area, and let's calculate how many molecules can be transported by this in 1 hour. So the surface area is going to be 6 times 10 nanometers squared, which is 6 times, well, what's 10 squared? 100 nanometers squared. So that's a total of 600 nanometers squared. Now, remember, it can transport 1 molecule for every nanometer squared. So I can cancel out these nanometers squared, and basically I can transport 600 molecules in an hour. Now let's look at how much is needed by this cell. Oh, I made a math error, so let's cross that out. This membrane, remember, can transport 600 molecules in 1 hour. How many molecules does it need? Well, the volume is the length of the side, which is 10 nanometers cubed. What's 10 cubed? Well, that happens to be 1,000 nanometers cubed. And it still needs 5 molecules for every cubic nanometer. We can cross out our nanometers. So this cell needs 5,000 molecules per hour. Notice, it can only transport 600 molecules per hour. Can this cell transport enough molecules across its membrane in order to survive? No, it will not. So what this leads us to is we can look at a relationship between surface area and volume. So here I have for a hypothetical cube. If its length was 1 nanometer, it would have a ratio from surface area to volume of 6 to 1. Because it would have a surface area of 6, a volume of 1. If it had a length of 5 nanometers, now that ratio changes. Instead of being 6 to 1, it's 6 to 5. If our length is now 10 nanometers, then our ratio is going to change even more. It's instead of being 6 to 1 or 6 to 5, it's 6 to 10. And When we double that last one to get 20 nanometers, our ratio decreases even more. It goes down to 3 to 10. So notice our volume is increasing at a much faster rate than our surface area. This means if you kept making a cell bigger and bigger and bigger, eventually it will reach a point where the cell membrane cannot transport enough stuff into it to keep all of the cell alive. So this is just a graph that you may run across. It is showing the surface area and the volume versus the length of the side of a cube, and you can see that the volume is actually going to increase at a much faster rate than the surface area will. So what this means for us is that the size of a cell is limited by its surface area to volume ratio. A cell can only get so big before it starts hitting this limitation. Now, we obviously have some cells that are larger. There are a couple of ways that cells get across this. So around this. So for example, they can put infoldings in their plasma membrane and their cell membrane. That increases the surface area of the membrane. You can see that here in these thylakoids. You can also see that in this heavily infolded membrane of a cyanobacterium. They can also have a shape that maximizes their surface area while minimizing their volume. So if you look at this neuron, notice that each time you have this branching of membrane, you're increasing the surface area. Same thing here. This cell is a cell in the lining of the intestine. Notice it's got these folds. These folds are called microvilli, and they are basically these little finger-looking projections. They don't move. The main purpose that they serve is they increase the surface area of the cell so it can intake more of the nutrients that are inside the small intestine. So it's basically increasing the speed of movement of things across the membrane. So let's start talking about membranes now. Membranes are going to surround every single cell. Remember, sometimes they have a cell wall outside of them in everything except for animals and some protists. And they also surround all of the membrane-bound organelles in eukaryotes. It's a fairly thin layer, only 5 to 10 nanometers thick, and it's got 4 major components. One of those is the phospholipid bilayer. Hopefully that rings a bell from freshman biology. We're going to talk about phospholipids in more detail in this next slides. Then there are proteins that are stuck in the membrane, and these proteins have a variety of functions that we're going to talk about later. There are carbohydrate chains that tend to stick out from the cell membrane, and they're important for either identifying a cell or interacting with things in the environment of the cell. And then cholesterol is important as well. This is why I will tell you guys, if you're complaining about cholesterol, cholesterol is actually good. You need the right cholesterol, and you don't need too much of it, but you do need cholesterol for the health of your cells. So there are some classic experiments that were done to help determine the 3D structure of the cell membrane. Remember, this is all like super microscopic. You can only see it with an electron microscope, so not even the types of microscopes that we have at our school. What they basically did is something called a freeze fracture. They took a cell, they encased it in, some sort of liquid, they froze it, and then they used a knife to basically split apart that cell membrane. When they did that, that took this extracellular layer and the cytoplasm facing layer and separated them. And then they looked at this under an electron microscope. And what they saw were these two images on the right. You'll notice all of these bumps on the inside of the cytoplasmic layer. Well, those are actually the proteins that are sticking out. The kind of background layer that you see would be the phospholipids. And you can kind of see some holes in this top layer, but you can also see that some of the proteins got stuck in the top layer too. Another classic experiment that helped establish what's called the fluid mosaic model, which is our current model of how the cell membrane works, was really ingenious. What they did is scientists took a mouse cell and a human cell. Now, these cells have different proteins on their surface. They forced them to fuse together. And what happened is after they were allowed to sit for, an hour, they found that the proteins, the surface proteins that would mark either mouse cell or human cell, were all mixed up. If the cell membrane were unmoving, what you would expect is to find your fused cell with half human protein and half mouse protein. Instead, you got something like this where they're all mixed up. So because of this and the previous experiment, biologists determined that the cell membrane is a mosaic, meaning it's made up of a bunch of small pieces mixed together, in this case the lipids, the proteins, and the carbohydrates, and that it is fluid. Components can move around. Now, what I want to point out is that if you're looking at a cell membrane from the side, these are, your phospholipids. Stuff can move back and forth, or maybe like coming out of the screen and into the screen. Stuff cannot move out of the cell membrane. Remember, the phospholipids are held in place because they have these hydrophobic tails. We'll talk about that again, which will not mix with water. So this is just another image of the structure of the cell membrane, showing you how it is connected to the cytoskeleton on the inside of the cell and the extracellular matrix on the outside of the cell. Granted, single-celled creatures don't have an extracellular matrix, but they'll still have a bunch of proteins, carbohydrates, signaling molecules, adhering molecules on their outside layer, just so they can interact with their environment. So let's talk phospholipids, because their properties are essential to forming a cell membrane. They are, you can call them bipolar, though that's technically not the correct term. They have both a polar part and a nonpolar part. The polar part is hydrophilic. It will mix with water. The nonpolar part is hydrophobic, and it is repelled by water. So the tails that you traditionally see on a phospholipid, these are nonpolar tails. They're fatty acids, so they're primarily lipid, and they're not going to mix with water. This head that you see is the polar part. It's got a charged phosphate group on it, and that makes it polar and makes it easily interact with water. So when you arrange a bunch of these in water, in a watery environment, they will spontaneously form organizations where all of the hydrocarbon tails, the hydrophobic tails, are separated from water with the polar heads facing out. What that means is the kind of most prominent formation that occurs when you put a bunch of these phospholipids in water is they'll form this bilayer. So notice at the top here we have polar heads, and this is a watery environment. Here we have the nonpolar tails of two sets of phospholipids facing each other, and then here we have another set of polar heads facing water. So anything that is highly polar will not be able to get through this membrane. Anything that is too large and can't physically fit in the spaces between those phospholipids is also not going to fit. So ideally, your membrane needs to be flexible and should not be full of these phospholipids that are super tightly packed together, because you need for things to be able to move around inside of the membrane. For this reason, it's actually better and healthier if you've got more unsaturated fatty acids in your phospholipids. That will keep your membranes more fluid. So remember, when you have fatty acids that have double bonds in them, that makes these kinked or bent tails, and that's going to prevent these phospholipids from packing in closely. So let's talk about some of the membrane proteins that are stuck in the cell membrane. These membrane proteins are really important for determining the function of the membrane, and they can determine a lot of the properties of the cell that this membrane is covering. Each part of a cell, whether it's the internal membranes or the external membrane, is going to have its own unique combination and collection of proteins. There are two major classes. Peripheral proteins are loosely bound to the surface of the membrane. They can be either on the inner face or the outer face. And integral proteins usually cross the whole cell membrane. So they're going to penetrate or go into the cell membrane. A lot of them are going to be what we call transmembrane proteins, meaning they span the entire membrane. These are often proteins that will transport things, so move them from one side of the cell membrane to another. So one class of proteins that we're going to talk about when we do a little bit with cell communication and talk about the nervous system, we'll talk some about channel proteins, which are specialized, I think of them like specialized doorways that will allow specific molecules or ions to get across the cell membrane. There's a very specialized channel protein called aquaporin. Water, since it's polar, has trouble passing through the cell membrane. It has trouble getting through this non-polar area. So you have this aquaporin, aqua means water, that allows water to travel into and out of the cell, and this is what allows osmosis to freely happen. There are some disorders that are linked with malfunction of certain channel proteins. I have here listed, for example, cystic fibrosis, where you have a chloride channel that allows chloride ions to pass that malfunctions and causes health effects that can limit the lifespan of a person with that trait. Carrier proteins are going to interact with a specific ion or molecule and allow it to pass the plasma membrane. The major difference between them and channel proteins tend to have a hole that goes through them. Carrier proteins usually will take things across the cell membrane by changing their shape, and when they change their shape, they'll somehow change their shape so that the molecule that was attached on one side of the membrane gets spit out on the other side of the membrane. So a link disorder, some people, it's suggested, there's some research to support that some people who are not able to easily use their energy for sodium-potassium transport with a sodium-potassium pump may be a contributing factor to their obesity. There's cell recognition proteins. These are glycoproteins, so they're proteins with a carbohydrate or sugar attached. They're those Frankenstein molecules that are made up of more than one type of biomolecule. Here is the glyco part of this protein, and a very important set of the cell recognition proteins in humans is the major histocompatibility complex, or the MHC, which is basically how your immune system recognizes your cells as not foreign invaders. And any cell that's introduced with MHC proteins that are not recognized by your immune system will be attacked and killed by it. There are receptor proteins. They will link to a specific molecule, and then they will send a signal to the inside of the cell. So a link disorder, people who are pygmies, they don't grow quite as tall. It's different from dwarfism, but they have plasma membrane growth hormone receptors that will not interact with human growth hormone. So while their body is producing the signal to grow taller with the human growth hormone, their cells aren't incapable of responding to it. There are enzymatic proteins that are enzymes that just happen to be stuck in the cell membrane. We are going to talk about these quite a bit when we talk about cell respiration and photosynthesis. Junction proteins are going to basically very tightly hold together cells. You have different types of junction proteins, like what we talked about at the end of the last lesson on cell organelles when we talked about gap junctions, tight junctions, etc. So proteins' properties are going to anchor them in place. For example, a membrane protein is going to have a region that is nonpolar, and that nonpolar region is going to sit in the nonpolar area of the phospholipid bilayer, the cell membrane. Any parts of that protein that stick out or are exposed to water are going to be polar. This helps a protein fold properly and helps it keep its shape. So that's what keeps a protein that's stuck in the cell membrane from just popping out in one direction or another. It doesn't do that because let's say this protein did start to slide out, well, it would be repelled by water, the nonpolar regions, and basically be pushed back into place. So here are the complicated shapes of a couple of different proteins. Notice we've got some interesting secondary structures here, secondary structures like the beta-pleated sheet and the alpha helices that you see. So those alpha helices on the molecule on the right, which is a proton pump, which pumps hydrogen ions into or out of an area, are nonpolar, which helps them stick in the nonpolar part of the cell membrane. So let's look at those carbohydrates that are attached. So they're mostly found on the outside of membranes. There are a lot of signaling molecules. There are two major types, glycoproteins, which are basically protein with some carbohydrates attached, and then there are glycolipids. So they will look like a phospholipid, except instead of having a phospholipid head, they'll have lipids coming out instead. They're important for recognition between cells and also for basically identification of cells. So here you see a glycolipid. It's basically carbohydrates coming off that are attached to some hydrophobic tails that stick in the phospholipid bilayer. So these are also important for signaling between cells, cell communication, and it's also really important for development. You have cells that are moving around in the body, and part of the way they know where to go is by these extracellular proteins, or outside the cell proteins and carbohydrate groups. Now, last but not least, cholesterol is very important. It is nonpolar, so it does have an OH group that is polar, and this can allow it to basically end up in the same area as the polar heads of a phospholipid. But the important purpose of cholesterol in cell membranes is that it will cause the cell membrane to become more fluid. And that's important because when temperatures get hot, the cholesterol actually helps to stabilize the membrane, keeps it from getting so fluid that you basically ooze into a puddle. And at low temperatures, it helps prevent the cell membrane from freezing. So it's also important for helping us maintain homeostasis and survive hotter or lower temperatures than normal. So let's talk permeability really quickly. The cell membrane is permeable to a very few substances. Permeable means it allows them to pass through. So for example, one example that I use is like a door into or out of a classroom is permeable to students. It will allow students to enter or exit. Is it permeable to cars? No, the doors on our classrooms are not big enough for cars to go through. They are impermeable to cars. So our cell membranes are permeable to small and non-charged molecules. Things like oxygen molecules, O2 or CO2, some alcohols, glycerol. They are impermeable to molecules that are large, starches or proteins, or molecules that have a charge, anything that's polar or an ion. Like I said, there's aquaporin, which allows water to enter into a cell. And if you need to get ions or polar molecules into or out of a cell, there are often carrier proteins or channel proteins that will allow that to happen. And when we talk a little bit more about membrane transport, we'll talk about how we get large molecules into the cell. Basically, what I say to get, if you want to get into the membrane, you cannot be large or in charge. If you're large or charged, you cannot pass through the cell membrane. And just another few notes on the extracellular matrix. We talked about it a little bit in the last slide, but I just wanted to give a few more details. It has a number of proteins that are outside of the cell that each have a particular job. We talked about integrin and how integrin helps cell attach to the extracellular surface. There's also collagen, which is going to resist stretching, elastin, which actually provides stretching, so it gives you some flexibility, and proteoglycans, which help regulate what molecules can pass through the cell, extracellular matrix and get from cell to cell. So there go tonight's notes. You will get more notes on the cell membrane later.